All right, guys, this is the first of two parts on dihybrid crosses. So what do we mean when we say a dihybrid cross? A dihybrid cross is when we're going to look at inheritance of two traits. So dihybrid cross is inheritance of two traits. Now, we said back in the last video that the law of independent assortment says that any possible gamete combination is as likely as any other. So for example, if we go back to our monohybrid, if we have a mom who's heterozygous, we cross that with a dad who's heterozygous or a male, then the mom can give either this, do a check mark here, this, or the dad can give that or that. And then this would result in a 2x2 box with the gamete, possible gametes at the top. But if we're looking at two traits, it can be a little bit more complicated. So let's look at a couple of examples. Let's look at example one. So let's say, and I'm not even going to give traits to this, I'm just going to use the letter representation. So let's say we have a female who's homozygous dominant for two traits. And we're going to cross that with a male who's homozygous recessive. And I want to know what their offspring would be. Well, hopefully you see that this is a pretty easy problem because I would be looking at the mom would only be able to give a dominant A and a dominant B, and the male would only be able to give a recessive A and a recessive B. So if we did a cross, this is the only possible gamete for mom. This is the only possible gamete from dad. So technically, it would be a 1x1 box, although we really don't need that. And what we would see is that all of the offspring would be heterozygous for both traits. I'm going to do a quick focus. I think I'm a little blurry here. That's a little better. All right, let's look at the next example, the next possible example. So again, like this could be, curly hair and it could be eye color. So it could be anything we wanted it to be. All right, so let's look at example two. What if I had a homozygous dominant for one trait, mother, and a heterozygous, and the dad was heterozygous for the other trait and homozygous dominant for the second? Okay, take a second and think, what would be the possible gamete combinations that I could get from mom? And remember, the offspring are going to inherit one A allele from mom and one B allele from mom, and one A allele from dad and one B allele from dad. Pause the video if you need to. Okay, did you say that mom could give the only type of A allele she can give is a capital? So she's going to give a capital A no matter what, but she can give a or a dominant B, or she can give a heterozygous B. And then dad, conversely, can only give a dominant B allele, but he can give either a dominant A or a recessive A. So mom has 2 possible gamete combinations. Dad has 2 possible gamete combinations. So how big of a box am I going to need on this? I'm going to need a 2x2 when I do my Punnett square. So I put mom's alleles at the top, homozygous. I'm dominant for both alleles. And then I have a dominant and a recessive. And then conversely for dad, I have capital A, capital B, and I have lowercase a, capital B. So now I'm going to draw these down just like I would if I was going to do my monohybrid square. But in this case, I've got an A and I've got an A. So let's just underline these. So I'm going to have 2 dominant, homozygous dominant, and homozygous dominant. Here I'm going to be homozygous dominant and heterozygous for trait 2. Here I'm going to be heterozygous for trait 1, homozygous dominant, and here I'm going to be heterozygous and heterozygous. Now, I have the same phenotype, right? Because everybody has a dominant allele for A, and everybody has a dominant allele for B. So my phenotypes are 100%. But my genotypes, I have 4 different genotypes. So I can be heterozygous for both. I can be homozygous for both, homozygous for one, heterozygous for the other, and conversely. So I have 4 genotypes. Okay, but this one a little more complicated than this one. But what I have to do, and here's the point to this, is you have to look when you're doing more than one trait, you have to look at mom's genotype and dad's genotype, and you have to figure out, okay, how many possible allele combinations can I make? So if we move on to the third one, and we're kind of moving up in the world here, we're going to get a little more complicated again. This time I'm going to cross a heterozygous by a heterozygous. Okay, let's look at one of these. How many possible gamete combinations can I have? Go ahead and pause the video if you need to. Okay, so I'm going to do a little bit of an arrow work here. So I could have a dominant A and a dominant B. I could have a dominant A and a recessive B. I could have a recessive A and a dominant B, or I could be homozygous recessive. And because dad has the same genotype, he's going to be able to give the same gametes. That means when he forms sex cells, he's going to give one allele to each of the sex cells. These are all of the possible combinations that he could have. And we have to consider that because the law of segregation says we do. Well, when I go to do this Punnett square, what do I have to do? Ooh, this guy's going to have, so I've got 1, 2, 3, 4. 1, 2, 3, 4. So I'm going to have a 4 by 4. Now, each gamete goes at the top, each possible gamete combination. Now, we're actually going to do this in the next video. We're not going to work this out now. We're going to look at some traits. And we're going to do some practice dihybrid crosses of this nature in the next video. And what I would do then is I would pull this trait down and this trait, or this gamete down and this gamete over, down and over, and I would fill in the boxes. So this one takes a little bit more work. But a lot of times people will say, well, do a 4 by 4 box. Anytime you're doing a dihybrid cross, well, if I did a 4 by 4 box here, I'd have this across the top. I'd have capital A, B, capital A, B, capital A, B, capital A, B. I'd have lowercase, 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 lowercase. It's a lot more work than you need to do. Or they'd say, do 2 capital A's, capital B's, and 2 capital A's, lowercase b's. Again, it's more work than I need to do. What you really do need to learn how to do is how to analyze the possible gametes you can get from parental genotypes. So here is, I'm going to write this in red because here's one of the big talking points for this video is that you have to have one copy for each allele. You can't have 2 A's, you can't have 2 B's. The offspring, let's say one of these is the number of legs, and let's say one of these is your number of eyes. You can't get 2 genes for legs and 2 genes for eyes. You have to have 1 gene for legs, or 1 allele for legs, 1 allele for eyes. So it's not going to work otherwise. All right, so here's a little bit of a thinking question. And then I'm going to show you the result. What if I was going to do 3 traits? A, B, B, C, C, and I was going to cross that with A, B, B, C, C. Okay, take a second. See how many possible allele combinations you could come up with and tell me how big of a Punnett square would you need to do? Okay, welcome back. Did you give that a shot? All right, I'm going to share my screen and I'm going to show you what that would look like. So Here we have all of the possible combinations, and you'll see that my box is 1, 2, 3, 4, 5, 6, 7, 8 by 1, 2, 3, 4, 5, 6, 7, 8. I could have all dominant genes. I could have capital A, capital B, capital D. Same thing over here. At the end, I could have all recessive, lowercase a, lowercase b, lowercase c, lowercase a, lowercase b, lowercase d. Then I could have dominant, dominant, recessive, dominant, recessive, dominant, dominant, recessive, recessive, so on and so forth. When you do the cross, you're going to pull in the A from here, the A from here, B from here, the B from here, D and D, so on and so forth. Same thing here. The A's come in, the B's come in, but here I pull in a recessive D. Here I pull in a capital D. So when it's all done, my phenotypes, the way that they're going to look, I'm going to have 27 of my, so let's see if we had 9 times 3, that would be 27. Oh, wait, this is the total, I think. Let's see. Got 1, 3, 6, 7, 10, 68. Yeah, okay, so that, that's not the total. Out of my total here, 27 would have this phenotype in red. So you see the red? 9 would have that phenotype. 9 would have the green. 9 would have the purple. And you can see it gets a lot more complicated. And if we did 4 hybrid cross, holy cow, that would be a big Punnett square indeed. Now, there's actually easier ways. I'm going to stop sharing this screen. I can figure out how. There are easier ways to calculate probability than doing Punnett squares. So when we get into the 3 trait, the 4 trait, the 5 trait, we would do it with statistics. But for our case and for our purposes in a sophomore biology class, we're probably only ever going to really do 1 trait or 2 traits. So, but let's do a quick takeaway. So before you do a dihybrid cross, you have to do, and so this whole time what we've been doing is called gamete, I call it gamete analysis. So you have to analyze what gametes mom can give and dad can give. And then we write those gametes along the edges of our Punnett square. Okay, so join me in part 2 where we're going to look at some examples of how to do this.